Hello, everyone. Thank you so much for joining us. We're really excited today to talk about cancer and how it impacts epigenetics. And as you know, so many people are getting cancer, and then you probably know someone who's close to you, or family or friends who might have cancer, or know someone who has cancer. So this is a really interesting topic, and we're going to be joined today by Dr. Joe Zundell. Dr. Zundell is a cancer biologist and postdoctoral researcher at University of Pennsylvania. And Dr. Joe is doing some really fascinating work in cancer therapeutics, and had a really great post recently about how cancer cell influence gene expression through our enzymes, and how targeting these enzymes can maybe an effective form of cancer therapy. So we're going to discuss today like how cancer spreads and then what happens on a biological level, and more about Dr. Joe's approach to targeting these enzymes for cancer therapy. So I saw.
Hey,
Hello, hi, Dr. Joe.
hey, how's it
How are
going?
you?
Thanks for having me. No, it's okay. I'm good. How are you?
Good. You made it in time. I was like really worried, and
Yeah,
thank you so much for giving us a heads up and then make it home.
absolutely. Yeah, no, I was just driving home, so I wanted to make sure I made it in time. But again,
Thank
thanks
you so
for
much.
thanks for having me.
Oh, likewise. This is like such a fascinating topic, and I just mentioned that we know so many people who have cancer and are developing cancer. We can see this is like one of the really like high prevalence of different kind of cancer, and we all know someone who has it, so like really important topic.
Yeah.
But tell us a little bit about your background, like how like you became a cancer biologist, and who's especially specializing in cancer therapy and also drug discovery.
Yeah, absolutely. So I mean, I've always been interested in science. If I wasn't a cancer biologist, I would be a marine biologist most likely. Yeah, I've always been interested in the ocean, but based on my upbringing, I didn't really have that many opportunities to do marine biology. And even though I initially went to school for marine biology, things weren't working out, largely due to funding reasons for marine
Hmm.
biological research. So I eventually got into cancer research, and a lot of this is personal, largely because my mom had esophageal cancer towards the tail end of my high school, and I kind of began emotionally seeking answers. A lot of the times, even when cancer patients get sick, they look for reasons why they're sick. And so from my perspective, I was trying to find reasons why my mom was sick and basically dying. She eventually passed away after about
I'm
a 2
sorry.
year struggle. No, it's okay. I mean, it kind of made me into what I am today. So through those tough experiences with my mom, essentially, it stimulated my interest in cancer biology to a very charismatic and passionate level that helped me push through the challenges of achieving an education in cancer biology. So a lot of it is personal, but also I realized throughout the various degree of schooling that I've had that I'm very good at like biochemistry and various wet lab techniques and things like that too. So they all kind of played a hand in how I became a cancer biologist today.
But I also felt like you're not just a biologist, you're also a super effective communicator, because so many scientists don't really know how to communicate that information yet. Like you're here, like really educating everyone about this, about cancer biology and mechanisms and everything around it.
Yeah, no, I really appreciate that. And again, I imagine a lot of people are very similar in mind to how I was trying to deal with my mom having cancer. And oftentimes when you talk to oncologists, they just blurt out the jargon that they've learned in medical school, and they don't really explain, even on a simplistic level, like how the cancer is working and how various cancers differ. And this lack of appropriate explanation leads people to seek alternative cures or remedies for their cancers, and oftentimes they make people way more sick, um, as I'm sure you have seen it, and many people here. So that's why communication is super important to me. This stuff is scary, and I know that it's very, very challenging, but in trying to break it down as simply as possible, it's giving people a logical understanding of how this disease works so they can have, separate fact from fiction, so to speak.
So what does a cancer biologist do? What do you do like in your day-to-day life?
Yeah, no, that's a good question. So a lot of it depends on the labs that we choose. So as an example, through graduate school, you pick a rotation lab or rotation labs. And so you pick multiple labs, you rotate in all of them. And generally you try and pick labs which have kind of diverse topics that you're interested in, but will give you an education in a bunch of different, types of cancer biology using a variety of different methods. So like as an example, I rotated in a melanoma lab. I rotated in an ovarian cancer lab where I ended up staying. And yeah, that's pretty much it. So I ended up staying in the ovarian cancer lab. And so when we commit to a lab, we utilize a variety of different techniques to answer questions. So it's all wet lab work. So it's all like, looking at what proteins are increased or decreased in cancer cell lines, in context of enzymes, which enzymes are more hyperactive, basically, which can induce specific characteristics about cancer cells, which make them worse than others. And obviously it varies between different cancer types. So ultimately it's just a culmination of just like wet lab. I don't know how familiar the audience here is with like technical things. So like using what's called a Western blot to look at protein expression between cells, or looking at how DNA is essentially read to produce RNA. We can do what's called RNA sequencing or qPCR to look at the total RNA that's made, and that can be representative of how a particular gene of interest is expressed and which may relate to a particular cancer. Throughout my thesis work, I focused on epigenetics. So that's why my epigenetics post was probably more clear than others, because I spent the past 6 or so years really digging deep into, what epigenetics is and various mechanisms through epigenetics, which can drive the specific type of ovarian cancer that I was studying. So there's a variety of assays which come with, or experiments which come with, examining epigenetic mechanisms for cancer.
Which is what we're going to talk about today.
Yeah.
Because you had a super informative post about how cancer can impact DNA, which is part of epigenetics. Could you this is super fascinating. So walk us through the steps, like how does, DNA is formed, and how is it stored, and how does it get impacted by cancer?
Sure. So again, I can't emphasize this enough that every cancer is different. So depending on the cell type that the cancer originates in, whether it's ovarian, breast, lung, colon, some of these common cancers like prostate cancer as an example, depending on where the cancer originates, they're going to have different characteristics on the DNA level. So DNA is in the nucleus, and it's essentially the directions for how cells go throughout their particular functions in their specific environment. So of course, cells within the prostate are going to function very, very differently than cells in the lungs. And so those environments, through epigenetic regulation, will dictate what sort of genes or segments of DNA are read over time. And sometimes, when somebody has cancer, there are specific genes, or let's say pages of a book, that are ripped out, or they don't work. You can't read them anymore because they're either mutated through a variety of different types of mutations, or there's even chromosomal deletions. So pieces of DNA that are completely deleted or create fusions. And a good example of that is actually, I live in Philadelphia right now. And I've posted about what's called the Philadelphia chromosome before, which is actually, as cells are dividing, the Philadelphia chromosome is essentially derived from when cells are dividing, they need to make more DNA. And in this process of making more DNA, when the chromosomes align at what's called the metaphasic plate in cell division, there's this crossing over event that occurs, and it creates a fusion between two chromosomes. and you essentially create a new chromosome, which is what we call the Philadelphia chromosome. It's a translocation event, is what it's called. And this fusion occurs at a very specific point on a chromosome, which eventually that DNA sequence ends up being read, and it creates a new protein, which is a fusion between an oncogene, which is like a protein which expressed highly or has high enzymatic activity, can induce characteristics which promote cancer, which cause cancer. And then another protein which regulates B cell signaling. So you have this oncogene protein attached to a B cell protein, and this drives characteristics in those specific cells to cause cancer in those cells by driving the growth of them. So obviously, it's very complicated. It's not always just mutations. Sometimes it's about yeah, I'm not going to get into too much detail. Just get on to other questions. But
Yeah. So basically, like when these kind of like gene mutations and other mechanism happens, they could make proteins, and then these proteins can then influence the cause of the cancer or also the development of the cancer, pretty much.
exactly, yeah, just to simplify things.
Yeah, so like really understanding how cancer occurs, like what impact it could then give us insight into like potential treatment, right?
Yeah, exactly.
So like,
Also, I do also have to emphasize that it's never just one thing, because there's this common misconception that we all have cancer and we're all dealing with it, but that's not true. Our cells, we have mechanisms within our cells to deal with these things should mutations arise. And so in order to cause cancer, it's more than just one event that causes transformation or the induction of a specific cancer type. So it's got to be two or more to actually induce specific characteristics where your cells can't fix it anymore. So just wanted to be clear there as well. So
absolutely. We often talk about, your DNA, your genetics is important, but many times genetic is not everything. So
absolutely.
your other factors like environmental factor, which actually impact epigenetics or your food, your diet, can actually pull the trigger. So we'll say, gene loads the gun, genetic loads the gun, but environment pulls the trigger. So that could combining with all other factors that you pretty much talked about.
Yeah, I think that's a pretty accurate statement, especially given, what we understand or what we've come to understand about lung cancer development. And there was a really beautiful paper which outlined the epigenetic transformation that comes from cigarette-induced lung cancer. So there was this beautiful paper in Cell published back in like 2018, I believe, which, essentially these biologists, they gave increasing doses of essentially a cigarette smoke concentrate into these cells, and they tracked the specific epigenetic markers over time, and they related that directly to eventually leading to an increase in the mutation of a very potent oncogene called KRAS. So they tracked the epigenetic changes that essentially led to the development of an oncogenic mutation, which is really hard to do in the context of wet lab biology. You need to be very careful, and that paper outlined just how important environment is in or can be in promoting cancer. Obviously, it's not everything, because there are genetic predispositions that people are born with, which can predispose them to specific cancer types.
Yeah. So you mentioned about like a role of enzymes, right? Because then if certain enzymes in the body can also impact like gene expression. So how do they do that?
Yeah. So there are a variety of ways in which you can do this. Either a cancer cell can, let me start back from just how adaptive cells are to specific environments. So as an example, I start smoking or I eat food or anything, normal or abnormal, whether it's smoking or just eating food. Our cells or specific cells will respond to that stimulus. And over time, if we kind of mess with that balance, the cells will adapt accordingly. And man, I don't know how to explain this super simply. Um, I even lost train of thought. Look at that. Yeah, I'm not perfect. Obviously, it takes a lot of time to be able to think about these things, but sorry, can you repeat the question again?
Yeah. So, like, what kind of role does enzyme play in the body, and how can they impact like a gene expression?
Yeah. So breaking this down again, sometimes it's about the abundance of an enzyme, but it also can be about the activity. And so in adapting to how our environments respond to specific stimuli, depending on the environment, enzymes can produce specific things, which eventually cause a cascade of events, which can lead to things that promote tumor cell growth. So it could be producing a substrate. So a common example of this, at least nowadays, is at least in ovarian cancer, which is what I used to study, is the overexpression of an enzyme called glutaminase. And glutaminase 1 specifically will utilize glutamine, and it will break it down to use glutamine as a fuel source to enter the citric acid cycle to continue essentially fueling cell division processes or cell growth processes. And so that's one way that enzymes can contribute, is by kind of rerouting fuel sources. Because, cancer cells, let's say they run out of sugar, they need another way to maintain their energy. So they'll use something like glutamine. And another way is, as an example, like you'll have gene mutations, a common oncogene like AKT or PIK3 kinase. Again, in ovarian cancer, PIK3 kinase, the gene is mutated. It doesn't necessarily always impact the protein expression, so you don't necessarily see an increase or a decrease, but this mutation causes the catalytic site of the enzyme to be super effective. And so it increases its catalytic activity without affecting the increase in expression or decrease expression of the protein. So those are some ways that cancer cells can utilize enzymes to support their own growth. Yeah.
So then, they almost have this like coexisting like relationship, right? Like a cancer and also like enzymes. So like we see how different enzyme activity can impact like a cancer growth. But how does like a cancer kind of like to like manipulate these enzymes for survival?
So either it could be through mutation, like I said, you can have a mutation that results in like increased enzymatic activity of an enzyme, or you can have increased expression. So like it depends on the level of the enzyme's ability to act on its specific substrate. So like glutaminase 1 and glutamine, or you can increase the expression of an enzyme, which will sometimes enhance, by having more of an enzyme, you can, contribute to a cancer in that way instead of, just affecting the catalytic activity by mutation. Did that answer your question?
Yeah. Okay. How does that, so then like, to translate to something more like actionable for people, like how does this work in terms of potential cancer therapy using like enzymes, and how do you like target them? How do you use it?
Yeah, and that's a great example. And I'll bring it back to the Philadelphia chromosome again, because it's a very common thing that we learn about in cancer biology. In fact, it's one of the first things we learned about, just because cancer cells, in adapting to their environment, they've essentially produced a new protein that is unlike other proteins. And so by knowing this, we can produce molecules to target this new enzyme and be more selective to cancer cells as opposed to normal cells. Because when you give this drug systemically, you're going to have off-target effects. But if you have a specific thing that's made by the cancer cell and you can produce a drug against that, it's going to be very effective if you have that ability. But most of the time, we don't have that ability. That particular drug, which targets the protein produced from the Philadelphia chromosome, is called Gleevec, or imatinib, which most people might know. And it targets the enzymatic activity that's essentially produced from this fusion enzyme that's created. So if we know the contributing characteristics of a tumor or of a specific enzyme or any molecule, really, we can produce a therapy, whether it's a small molecule to target the catalytic activity by competing with the material that the enzyme normally uses in functioning, or we can, as we're getting into now in modern science, biologics, using things like antibodies to kind of promote our immune systems against tumor cells. That's a completely different topic than what we're talking about. But yeah.
Well, we can kind of see with with the COVID vaccine, it's already kind of like gearing towards that direction, right?
Absolutely, yeah. So we are, well, not me specifically, but actually, I know for a fact that Moderna is working on mRNA-related therapies for various cancers. I'm actually assuming we'll find out something within the next half a year to a year about what they're doing. Same with Pfizer, actually, too.
Oh, wow.
Yeah.
So this is seems like the future, right? If we have targeted therapy, then people won't have to, because one side effect from all the chemo drug is it kills everything. It's not just the bad cells, but it actually kills everything. But now with these targeted therapy, then your good cell can still live and you just kill the cancer cell, which then less side effect and then more effective.
Absolutely. And I will say also that all the therapies that we produced up to this point have specific targets. So they are technically targeted therapies. But I think what we want to be careful of is saying that they're not targeted because we design all these drugs. It's not like they just have a random effect. They're designed to target specific enzymes. But the difference comes with, the differences between people of how cancer patient will respond to a drug. So what's really important here is how do we personalize a targeted drug to make it more effective for a specific cancer patient? And that's something I think, because of just how complicated cancer is and our current screening methods, it's always going to be something that we struggle with. But personalized therapy is something that we're always trying to do better on.
When do you think this will become prime time? Because I know it's also really expensive, right? If you really studying this particular person's genetic, epigenetic, and then having to make a drug just for this person, I would see that still extremely costly. Maybe not everyone can afford it. When do you see it will become prime time?
Yeah, so it's again, we have these therapies, but it's not that we can't, it's not that we necessarily need to design them. We know enough about many different types of cancers to have therapies for them. They're already even in the clinic. But it's important we know what makes something a personalized approach is using the right drugs. Yeah, not necessarily, the standard of care. Yeah. So in answering your question, I don't realistically think we're ever going to have a reasonable personalized strategy just because, again, of how complex cancer is. And I'm not being a pessimist by this, but it's, like you said, I don't think anyone reasonably is ever going to have enough money to, unless you're, Jeff Bezos or Elon Musk, you just throw your money at your problems, truly pay for a personalized strategy to your specific cancer. At least not within 10 years. It's unfortunate, but perhaps one day we'll get there in my lifetime, but it's a huge challenge, and it's something that we've been working on for the inceptions of cancer biology, how to make something truly personalized and effective for each patient so we can maximize the efficacy of a particular drug and reduce mortality.
I guess minimally, at least if people understand that this is a possibility and also understand their own cancer a little bit better, at least they can ask the right questions with their oncologist, okay, for my cancer, my physiology, my genetic, what's the right drug that I should use to target this particular cancer?
Yeah, definitely. And that's something that I try. So on my page, I can't legally give medical advice because we can't do that on Instagram. But I feel that and making things as clear as possible, and kind of showing people how we produce drugs, and explaining, with my little pictures how, what a drug is targeting and how that technically works as simple as possible. It gives people questions to ask their oncologist, like, hey, will an HDAC6 inhibitor be right for me? I saw this post which explained that it worked this way by blocking this enzyme. Or if I have the Philadelphia chromosome, will Gleevec work for me? Actually, there was a new drug that was made because cancer cells were developing resistance. Remember how I said cells are very adaptive? Cancer cells are even more adaptive to our environments than our normal cells because they're kind of fucked up, for lack of a better word. Use my language. But they've developed resistance to a very important drug, Gleevec, which was developed a while ago. So we've recently made a new drug because they developed resistance called Semblix. And so instead of, Gleevec binding to the catalytic domain of this enzyme, this fusion protein that's produced, they produced another molecule which binds to a different portion of that enzyme, which causes a change in the shape of the protein. So now you can give something like Gleevec and this other drug, and they'll both kind of interact with this protein to further reduce its activity. So, yeah, that's kind of how we go about those things.
Makes a lot of sense, because I also think, like, you're kind of like in the frontier of this research, what's going on. And I felt like many oncologists, they actually might not be up to speed with all these like different reaction, new drugs and all these things. So when a patient actually understand and know about this information, then they can actually ask their oncologist about the drugs and about, resistance and stuff, in case that their oncologist didn't even know.
Yeah, and I think, yeah, absolutely. And I think realistically, you can't expect an oncologist or a surgeon to not only not have the time to discuss with their patients, but also not have the education on it, because they've got so many things going on. I'm a specialist, so there's a reason that I know what I'm talking about cancer biology, because I do it every day. Just like you couldn't ask me, how to do surgery to excise an ovarian tumor from a patient who is 50 some odd years old and is exhibiting these particular characteristics. I couldn't do it. I couldn't tell you how to do it. So I do also want to emphasize that this is something that a lot of hospitals do, whether it's for cancer or for something like Parkinson's. As an example, we have disease boards. In the case of cancer, they're called tumor boards, where they'll get experts who understand a particular patient's, cancer, and they'll suggest, based on what they know, current clinical trials, what strategies can be effective for a given patient, which also does help to personalize things. But they can then utilize these specialists to discuss with a cancer patient why they're receiving certain things. I have a friend that works, he's actually a sales rep for Abbott. He does Parkinson's related work, and one of his jobs is actually explaining to his the hospital's patients, Parkinson's patients, how this particular brain implant works to reduce its electrical stimulation, which reduces characteristics which come with Parkinson's. So he directly interacts with the patients, opposed to them directly interacting with a surgeon, and he helps explain, how things work. And we do the same for cancer, but it's maybe not as prevalent as some other fields. Something we need to be better at for sure.
Yeah. We have one question came in. Hi, what's the mechanism of relapsing? Is it the same as getting cancer in the first place as a lymphoma survivor in remission for 5+ years? Are the odds of me getting cancer back the same as it was before?
Yeah, I'm sure you're going to expect this answer. It depends. Because I can't answer that question in good faith, because I don't understand all of the characteristics that went into driving this person's cancer. I also don't know their treatments, nor do I think I'm qualified to understand everything that went into this, because I didn't, examine this person over the 5 years or more of their cancer progression. But a lot of this will depend on how effective the drug was, how early staged the cancer was, and whether or not it spread to distant tissues. Because metastasis does complicate things very, very heavily. Um, so yeah, that's kind of my answer to that.
But it's a really good sign that this person is in remission for 5+ years.
Yeah, and I mean, an example of this too is, and it's kind of sad. Like again, going back to my mom, she had esophageal cancer, and she went into remission. Um, but you know, like I said, every cancer is different, and some drugs work better for some cancers than others. She went into remission, and you know, sometimes specific drugs, despite their targets, they take care of most of the cells within a tumor, and they leave behind a very small fraction um, of tumor cells that have evolved in different ways, using different mechanisms. So that drug that you use doesn't target them. You killed off the essentially competing cancer cells because it's an evolving environment where they all compete for resources, even amongst cancer cells. You kill off the competition, and now suddenly you've left this population which doesn't respond to this drug. You've essentially given it some room to grow. Now they don't always grow, but in the context of my mom, it grew back quite violently, and that relapse was eventually what killed her. So if by now you haven't had, so this is to that person, if by now you haven't had some form of relapse, it's a very good sign. But it is super important to keep, checking up on doctor's visits. Yeah, because some cancers are easier to detect than others. But it's super important to keep checking in with the doctor to make sure that you don't have a relapse. And if you do, detecting it early is super important. But again, it's going to vary depending on tumor type.
Absolutely. You've done like a lot of amazing work, and then
Thank you.
Ovarian cancer is like something you're most passionate about as of now.
So I got into ovarian cancer just because I realized I was good technically at the stuff that I was doing in an ovarian cancer lab. They just happened to be studying ovarian cancer. I wanted to study, it doesn't matter what cancer I study. I'm interested in all cancers, and that's why I try and spread awareness on my page to all different types of cancers, even the rare ones. But realistically, so in my postdoc, I'm actually finished with my postdoc right now. I actually started a new job this week, but in my postdoc,
Congrats.
I'll talk about that shortly. But in my postdoc, I studied liver cancer. I was in Dr. Celeste Simon's lab at the University of Pennsylvania, examining liver cancer. And there's a specific enzyme that we studied in that lab, which is basically epigenetically silenced. The enzyme just completely is lost in the liver. And this is one characteristic which drives hepatocellular carcinoma or liver cancer progression, obviously over time, because it is a cancer that is frequently seen in people 60 or older. Mm-hmm. Excuse me. But that lab also studied pancreatic cancer and or PDAC, pancreatic ductal adenocarcinoma, and various types of sarcomas or cancers of the muscle tissue or bone, as an example. They also studied kidney cancer. So I do have like a little bit of background in renal cancers or kidney cancers, hepatocellular carcinoma, liver cancer, sarcomas. Ovarian cancer was my thesis work. Prior to that, prior to graduate school, I did a lot of work, and my first publication was actually in leukemia, a type of leukemia called, which is a blood cancer called chronic lymphocytic leukemia. So, so it's a lot of work. I wouldn't say it's amazing, but it's a lot of work. Obviously, a lot more to be done, but I'd study any cancer, so it's not specifically to ovarian cancer.
Seriously. Okay. Let's talk about your new job. You were just going to say something.
Sure. Yeah. So I finished my postdoc largely because I realized it wasn't for me anymore. I had this track to become a professor, and I wasn't super satisfied with how academic work is structured to become a professor. And that's, kind of saying it politely.
I can echo.
Yeah, so I kind of got tired with the academic structure, and I wanted to try something new. I wanted to, learn more high throughput ways and more translational ways to actually, help cancer patients. Because that's why I got into cancer in the first place, was to, study therapies which have a high potential to go into patients. And so I left my postdoc, and now I actually started a position nearby in Philadelphia at a biotech company called Insight. And there I'm tasked with the study of examining bispecific antibodies for a variety of different types of cancers. So it's a type of immunotherapy. Basically, we are using antibodies, and I've posted, I actually, if people are curious, I just did a post explaining what bispecific antibodies are, because I wanted to preface, my own transition and learn a little bit. But essentially, it's priming the immune system against tumor cells using specific markers. I can't talk about the specifics because, there are just disclosure agreements that I can't talk about that. But I can talk about the overall platform for the general biology. So
That's worth another Instagram live. Like, we're gonna have to invite you come back to talk about immunotherapy, because that's like a whole other topic.
yeah, and so like I've been covering. So I started another series on my Instagram page with Yasmin Mohseni. Her Instagram handle is at Dr.Yas YAS_. And we've been doing a cancer immunotherapy series. Just like, what is immunotherapy? In the context of cancer, how do we go about producing an immunotherapy? What are the various types? What are the ones that are already FDA approved for specific types of cancers? Those sorts of things. So, we'll definitely talk more about that in the future.
Amazing. Wow.
Yeah.
Thank you so much for what you do and spread the knowledge and then educate the public about all these things. It's just so important.
I appreciate it. Thank you so much.
Thank you. And then we have a lot of people joining. So like, everyone, please follow Dr. Joe at Dr.Joe Zundell. We'll have everything in a post promoting this Instagram live. So thank you so much.
Yeah, no problem. And I think, are there any questions? I know that people have the ability to ask questions in the.
Let's check again. I think
Since we have some time, we might as well answer some questions.
Okay, actually, yes, there's one more question.
Sure.
Any book I can educate myself about these stuff? A scientist from different field, but curious.
Yeah, so the hard part about this is for cancer biology, and I don't mean to sound like superior in any way, to understand cancer biology, and this is why I try and simplify it as much as possible on my page. To understand cancer biology, you have to have a foundational education in a lot of other fields. So I can suggest, there's literally a textbook called Cancer Biology, and it's written by Bob Weinberg, who is, I think he's at Harvard right now. He's a very good professor. I met him a few times. I can suggest that Cancer Biology book written by Bob Weinberg. But realistically, it's going to confuse the crap out of somebody who doesn't have a really hardcore molecular biology background or biochemistry background, or even immunology background, even basic immunology, just, of how, we're coming to understand cancer progression. So all I can realistically say is maybe my page. I'm actually thinking about writing a book.
Please do.
Yeah, so it's been in the works. Obviously, it's going to be a culmination of my Cancer Bio Basics series that I've done on Instagram. I'm going to turn that into a book for laymen, essentially. So I don't know how much time that's going to take. It's still in here, and it's not even on my computer yet. But yeah, it'll come eventually.
We'll be waiting.
Yeah.
Another question is, how do cancer stem cells arise? Can they be targets for therapy?
Yeah, so a very hot topic amongst cancer biologists is cancer stem cells. But realistically, we don't know anything about, how, well, we have an idea of how they can contribute to specific things which promote cancer, but we don't have super strong reasoning due to the lack of abundant evidence. We have evidence, but it's not super abundant we can't make super strong conclusions about their contributions or even how we target these specific cell types in cancers. And a lot of that stems from their abundance within a tumor. They're not super common within a tumor, so they're very hard to study. So we can't really parse out just how important they are in driving characteristics which promote tumor growth. And I think that stems from, again, an assumption from people where they assume that cancers are just like one type of cancer cell. But they're not. Cancers, particularly solid tumors, in one type of cancer or a solid tumor, you can have a variety of different cancer cells at different stages in their evolution, and they compete with each other for resources. They compete with the immune system for resources, and it's this constant battle between each other and our immune systems for survival. So it complicates that kind of cancer stem cell work very dramatically. And as such, that's one reason why we combine particular therapeutics for a cancer as opposed to just single treatments to kind of target those multiple varied populations of cancer cells that exist within specific cancers. Yeah.
That makes a lot of sense. We have one more question.
Sure.
Are chemicals in cosmetics safe, allowed dosage? I don't know. I don't know if you have any thoughts on that. Otherwise, I can probably help answer that question.
You can, because you've studied these things more than I have. But from my own perspective, and then you can provide yours, I think, certain chemicals may produce issues. The degree of which that they will or contribute to cancer, the degree of which they'll contribute to cancer is highly variable, just like anything in life. But I was actually just reading something recently that there's a potential contribution between like chemical hair straightener, which a lot of women use because of, beauty standards, which are seemingly unrealistic. Don't even get me started on that. But yeah, so like as an example, consistent and chronic use of chemical hair straighteners can likely contribute to I think even ovarian cancer was the paper.
Uterine cancer.
Yeah, uterine, my bad. Oh, so you read the same thing. Yeah.
Yeah, I read the same thing.
Yeah, so maybe you can provide some more insight on various other things.
So basically that's kind of like what we do, and we're trying to ask people to reduce their environmental exposures, which then can potentially maybe have less interaction with cancer cells or other things. The issue, even though we think all the cosmetics that we use or any personal care product, they should be under, allowed dosage or allowance, how much chemical is in it. However, we never have the causal study. We have enough causal study in animal models, but we don't have enough in humans. So we actually don't know. The issue is that so much epidemiological research have demonstrated that even at low dose exposures, even though you just expose yourself a little bit, but you have this day in and day out exposure. So over time, you actually being exposed, and then that causes a slew of health issues. So don't trust that any product that you're using that's actually safe. It's really, really important for people to understand that, to avoid certain chemicals, because these chemicals over time, these exposure can cause issues. So I wouldn't trust just say any product's safe. So this makes reading label, understanding, particularly avoiding these hormone disrupting chemicals are really, really important.
Yeah, I think we have to also be very careful of when we use words like detox and hormone disrupting, such that we communicate, specifics of what types of chemicals and what actually the literature is saying. So, the National Toxicology Program, at least in the United States here, they're always updating what are carcinogens, or what do we know about specific doses of carcinogens. So something can be carcinogenic, but realistically, if we're not associating with a toxic dose to induce tumor formation, realistically, we're going to be safe. But again, the data is quite variable. But I think this is one thing that I really like about your page, is that you're trying to promote awareness about specific types of chemicals that the literature is showing to be potentially carcinogenic, or not even carcinogenic, just maybe contributing to other health issues like allergies. And that's
absolutely
that I've collaborated with other people on talking about as well.
Absolutely. Hair straighteners, like hair relaxers, essentially, pretty much.
Yeah, they're essentially chemical denaturants. So your hair is a bunch of keratin proteins. And, you know, everyone, depending on their genetics, the way that their scalp is configured will affect the way that hair curls, or, you know, the shape of hair. And so these chemical denaturants, they act like they relax the proteins in the hair to make the proteins more straight. Chemical straighteners.
I would say if you can avoid, definitely avoid. You don't need to straighten your hair. Well, this is a whole separate topic about.
Well, that's what I said.
Which we can talk about.
Standards, yeah. So my wife has beautifully curly hair, and I love it. And so it always frustrated me when, you know, that at least in the beauty industry, that a lot of people just push this straight hair image if you know, if people have curly hair. But I don't know. I've always really appreciated a good head of curly hair.
It's funny. It's like I have straight hair. I always wanted curly hair, and all the people I know with curly hair, they wanted straight hair.
Yeah, I guess it's kind of wanting what you don't have.
Exactly.
But I can say that I admire her curly hair, but I don't want it. That's a lot of work, man.
It's a lot of work.
I mean, I can't imagine, you know, straightening it, but you know, having curly hair is something else. I like laying on it though. It makes for a great pillow.
Awesome. So thank you so much for staying long and answer everybody's question. And we'll be following you, and we're gonna have to invite you back to talk about immunotherapy, and I'm gonna keep up with your work and your book.
Yeah, absolutely. Yeah, no, and I'll definitely keep people updated on my page about, you know, kind of the things I'm learning, and try and explain them in layman's terms for those that follow me. So thank you again for your time. I really appreciate it.
Thank you so much. Talk to you soon then.
Yeah, see you, Jenna.
Bye.